thank you for joining us because citizen-driven research is the way that we can get things done. And with that, I'll turn it over to you, Christian.
Great. Well, thank you. Thank you, McFinn, and thanks everybody else for joining. This is a special, different kind of format this evening. As if you saw today, we actually shared some materials even ahead of time so that people could take a look at what we were going to cover. And tonight or today, depending on where you are in our time zones, we are joined by two of our longtime true expert physicians and community members who have always been here to help answer questions and to help guide some of the conversations when there are questions exactly the kind that we're going to cover in this program, which have to do with the science and understanding what's really the message behind some of the scientific communications that are out there. So joining us for this program are Dr. Terry Heiman-Patterson and Dr. Lyle Ostrow. So I'm going to give a quick intro for those who are not as familiar with them, and then we'll be turning the program over to them. So first, Dr. Terry Heiman-Patterson is the neurologist and ALS specialist who leads the MDA ALS Center of Hope at Temple University. And with a career spanning decades, she has dedicated her work to advancing the ALS clinical care, clinical trial implementation, and translational research to bring ideas from the lab to treatment in ALS. And to that end, she has led the studies on clinical care, genetic modifiers, biomarkers, pulmonary involvement, and brain-computer interface technologies for ALS. Dr. Heiman-Patterson is on the leadership committee for the Network for Excellence in ALS Research, NEALS, and has received the Forbes Norris Award, Lou Gehrig Award, and Tony Diamond Award in recognition for her contribution to ALS research and patient care. And she's the president and co-founder of ALS Hope Foundation, a nonprofit organization dedicated to making a difference in the lives of people living with ALS. Joining Dr. Patterson tonight is Dr. Lyle Ostrow. Lyle is a neurologist and ALS researcher and director of the Neuromuscular Pathology Lab at the Lewis Katz School of Medicine and MDA ALS Center of Hope at Temple University. He serves on the scientific advisory board of right here at EverythingALS and is on the board of directors at ALS Hope Foundation. Dr. Ostrow is chair of programmatic panel for the Department of Defense ALS Research Program, the ALSRP, and the largest dedicated annual funder of ALS therapeutic discovery and validation. He's also director of the Temple ALS Postmortem Corps, which is funded by the CDC National ALS Registry to provide autopsy tissue samples and data to ALS researchers around the world. It's exciting to have them both here. So we're going to turn it over to them to get the conversation going and to walk through the format and the goals of the program to start us off. So Terry and Lyle, the show is all yours.
Okay, so yeah, thank you guys so much. We're really looking forward to this, and I wanted to talk a little bit about what we're going to do and how we've structured this. And for those of you who joined us last time, this is going to evolve, and we're still sort of learning how to do this and what works. And we want your input in helping shape that in the future. I'm going to quickly go over kind of what's our inspiration, what's the plan, why are we doing this, a couple ground rules, but the main point is what's in that green box there, right? So we need to work together to be responsible consumers of information. We live in an amazing time. So first of all, in ALS, there are a zillion therapies in trials, and it seems like there's a new trial or a new result we hear about almost every day, right? And it's impossible to keep them all straight. And I get emails every, day from people, what do you think of this? What do you think of this? And it's a never-ending, barrage of information. But that barrage of information is also part of what's so cool about this world we live in, because we find out about stuff really fast, right? And so things going on around the world, we have access to information like never before. And that includes science, and that includes, research papers and all that other information. And as you guys know, there's a lot of really cool, exciting stuff. There's also a lot of stuff that, while it's cool and exciting and potentially groundbreaking, sometimes gets overhyped or, maybe is very early in the process and not ready for prime time. And it's important that we all understand that. And it's also important that we understand, what makes good research. And so our goal with these sessions is not so much to sit here and, have you guys ask us, what do you think of this one? What do you think of that one? And by the way, we are going to talk a lot and have a lot of time for questions about whatever you want to talk about. But our goal is sort of to work together to make you guys comfortable assessing this stuff on your own. And the analogy that I think of a lot, so I don't know who on this call has ever had like a teenage driver in their house, but once upon a time, everyone in this country before cars, we all like people got around in horse-drawn buggies, right? And then cars came around, and cars were a lot faster and they were a lot better, and they got us where we needed to go much more quickly, sort of like information today, right? But cars also like crashed a lot, and people were terrible at driving them, and they blew up and they made a huge mess. And so somebody said, you know what? We need driver's ed. And we need to have a standard way that we teach people to drive, because even though cars are fast and they get us where we need to go, we're all crashing into things. And so in some ways, we're kind of hoping that this is like a driver's ed of understanding this kind of research information. So in these sessions, I want my slide advanced. Basically, each time we're going to start with a short slide presentation about something. Today, Terry's going to talk about how to read a research paper and go through some of that with you guys. In future sessions, we may review specific papers, stuff that you guys send in. We actually hope that as this goes along, that maybe some of you will lead some of these sessions and these presentations at the beginning. And then we're going to have every time plenty of time for questions, okay? Plenty of time to talk about whatever you guys want to talk about, with a few ground rules. So number one, we're not here to tell you which trials we think are more or less likely to work, because let me, like, little secret here, anyone who does tell you that, they're full of it, because we don't know. If we knew, we would have done a much better job over the last 40 years, okay? We don't know. We can tell you what makes a well-designed trial, right? We can tell you what's the rationale. We can talk about, for a given therapy, like, why are they testing this, okay? What makes a rigorous, well-designed trial so that we'll get to an answer. Those things we definitely can talk about. But anyone who tells you that one trial is better than another, we just don't know. That's why we do them, okay? We are also, as I said, happy to talk about some of the rationale, some of the science, some of the different strategies. We ask you, please don't use the chat or the discussion time to promote specific treatments or therapies, okay? That's not why we're here. This isn't the place for that. But beyond that, really anything you guys want to talk about. And there's a lot of topics that have already been suggested. I'm not saying we're going to do these or not. I just sort of wanted to, put them up here because there's going to be some surveys and get your guys' opinions, but some stuff that people have suggested maybe for the future, to have a presentation about how do we find therapies, right? Why do things get into clinical trials? Who decides? How is this stuff funded? How do we choose a target? How do you review a research proposal? Some of you on this call know that people living with this disease and advocates now are being asked more and more to serve on committees, like for the one I chair for the Department of Defense, and actually review research. So what's that process like, and how do you do it? How do you actually design an experiment to test a new idea? Like, what's that about? Yeah, I got a great idea. You read something. Oh, we should try this. How do you do that? Serving on patient advisory boards. These are the sorts of things that some people have suggested, and we're going to get your input to figure that out. So with that, those of you who know me know I talk too much, but Terry and I are good partners in this stuff. Oh, I don't know why it's whistling. Is that me? I don't think so. In any case, I'm going to hand it over to Terry, and she's going to talk to you guys about how to read a research paper.
As
I
Terry
think, Terry, you
coming
there?
on board, I just wanted to say what a gift this is, Lyle and Terry. This is something, I wish I had when I was going through with Peter. So thank you for making the time. We so appreciate you and Terry just coming in and openly talking to people. And, we are so grateful for you to do this, because a lot of people don't. They don't try to go away from their clinic. So this is
and do a real quick just advertisement too. Like, I join a lot of the fireside chats with the Pathfinders, and I'm not doing it to tell you guys things. I'm doing it because I loved it, man. I learned so much from you guys, but it's just another avenue where we can engage and we can talk about whatever you guys want. So do join them.
this is such a gift. So Terry, the floor is yours.
So, I'm going to go over, and Lyle, please jump in as you'd like to, as commentary to. We're going to talk about how to read a research paper, and And then we sent around a paper. I hope everybody got it. After we go over some general pointers about reading a paper, we're going to critique that paper. And, I hope some of you at least read the abstract. But this paper claimed a total reversal of ALS confirmed by various, EMG changes, structural reconstitution, neuromuscular molecular restoration using computerized brain-guided re-engineering of the 1927 Nobel Prize fever therapy. And this was a therapy using malaria to create fevers. But anyway, I hope that by the end of it, you'll really have learned as we go through some of the critique. But start with some general pointers. So, first thing that we think you need to understand is what's a paper made of. When you face a research paper, what are you looking at? Well, first there's an abstract, and that really is a summary of the paper. And, often it sounds pretty good if you read the summary of the paper tonight that we passed around. You'd be like, oh my God, let me line up. The paper itself should have an introduction and a background. That's where you find that information about what is the premise, what is the hypothesis, and what data is that based on? What's been published in the area, and why did you choose, to do the experiment or the clinical trial? Why did you choose the molecule you chose? What is the rationale? Then methods is simply, okay, I'm asking this question, I'm studying this drug. How am I going to answer a question if it's a research experiment? Or how am I going to do this trial if I'm taking a drug to do a clinical trial? And is there enough information given in the methods that if my neighbor wanted to reproduce the trial or the experiment, that they could go ahead and do the same thing and get the same results? The results, of course, are what is found in the experiment. Or I know that many of you or most of you are interested in clinical trials primarily, so that's why we always talk about it. What was the actual data that was collected? And then how do you interpret the conclusions and discussion, or the interpretation of the experiment you did? What were the results? And how do they fit into the grand scheme of things? Where do you want to go next? And what are the caveats? You should always have in there, what are the caveats about the way the experiment was done, and what problems or things could get in the way with the interpretation. And references are simply the relevant literature. What did you base your background on? And so, pointers are, think critically. Don't always assume that the authors know what they're doing. Okay, that's and you don't have to be a scientist. Just, does that make sense? And if you have to think about, does that make sense? That might be a red flag. Ask questions and think about, after reading the background, are the investigators really asking the right question, solving the right problem, approaching it logically? And are they making reasonable assumptions? You can read creatively, right along the edges of the paper. Think about, what are the good ideas? Can I translate that anywhere else in science or elsewhere? Take notes. And we sent you a guide, and you can use our guide. We sent out a guide. You can use the guide to ask the questions. You can take notes using our guide when you approach any other paper that you're interested in. You can underline key points and mark what you think is most important or most questionable. And then after you read through everything, then try to summarize it in a few sentences. And that's how we know that we understood everything. If not, then maybe you need to go back and reread what perhaps you can't understand or you didn't quite get. So, that's kind of just some pointers. And then, we did provide this kind of guide, and I'm going to use this to these major questions here. What was the hypothesis? What methods were used? What about the design of this? I don't know that you could call it a trial, and we'll get to that. And what were the results, and did it answer the questions? And some things about when you're doing a clinical trial, even if the N is 1, that you should be asking, and this was an N of 1. How do you interpret the results? We know how the investigator interpreted the results, and we'll go through that. And, what do you think comes next? And was there any issues, any caveats that were in it, in the description or in the paper? So let's go ahead and start to critique a total reversal of ALS. And this, of course, would be exciting to anyone with ALS. Oh my goodness, is this a real treatment? Should I go down and get this treatment to Florida, which is where it's given? So let's start with what was the hypothesis. And so the group of investigators proposed that by inducing a high temperature, hyperthermia, you were going to upregulate heat shock protein, and this would help to clear clumped up TDP, right? We all have heard that TDP is a protein that aggregates in ALS, and it's found in virtually all people with ALS, pretty much, not 100%, but pretty much 98%, and that it may be part of the problem. So maybe we can clean it up. We can upregulate heat shock protein, get rid of the TDP aggregates, and then the neurons would recover. And it was based on this malarial fever treatment for, syphilis, basically, dementia paralytica. And also the investigator in his background said, look, ALS neurons have elevated heat shock protein, and TDP aggregation is increased, and it was correlated to heat shock protein levels. So the lower your level, the more TDP-43 was aggregated. So there must be a relationship there. And therefore, if I do if I do hyperthermia, if I heat up the body with this method, well, I can get the heat shock protein up and decrease TDP-43 aggregation, and that would work. And they pointed out some animal and cell culture experiments regarding heat shock protein. And so their hypothesis was basically that this therapy, CBIT as they call it, which is computerized brain-guided thermofebrile therapy, very special, and you can see the apparatus that they show on their website, can reverse ALS through the induction of heat shock protein. So, face value, maybe that sounds good, right? So let's keep going here. So what methods were used to answer the question? Well, in a trial, we want to know how many people were enrolled. Well, there was only one person enrolled. So this is what we would call anecdotal. The trial was only 6 months, and we'll talk about that. There's no placebo, right? It's an N of 1, and it certainly wasn't double-blinded because the doctor giving the treatment and the patient all knew they were getting treatment. There was no inclusion or exclusion criteria. It was just anyone who would, I think, pay this institute. How were participants chosen? There's no way to know how this person was chosen. And how did they determine the dosage and frequency of the treatment? Well, that wasn't clear either, although they did have a strategy. And there was no mention of safety monitoring, and were the endpoints, we'll talk about that, appropriate? So let's go into a little bit of detail. So a critique of the methods that we've already started. This was not a trial, right? There wasn't a placebo group. There wasn't a test group. It wasn't double-blinded. And there's no way, not being double-blinded and of one, that you could distinguish between treatment effect, the variability of the disease, and you don't even know that the person they're describing was properly diagnosed, because the way they said about the diagnosis was it was made by a solid neurology clinic, ALS at the Mayo Clinic, and there was no information, for instance, about exclusion of mimics or the diagnosis, other than saying the patient was diagnosed at ALS with ALS at Mayo Clinic. And then somewhere else in the paper, they mentioned nystagmus, which is a shaky eye movement that isn't usually considered part of ALS. So I don't know that we could even know that this was a confirmed diagnosis. There was only 6 months of follow-up, which is really insufficient to determine if the improvements would continue. That is, especially as you remember, there can be plateaus in the disease. And the clinical measurements pre and post were performed. They did vital capacity. They looked at ALSFRS. They did a bunch of, biomarkers that we'll talk about. But some of them could have been influenced by spasticity. For instance, gait, and just walking will loosen people up. So, you don't know. And as I'll mention later, some of the biomarkers like NFL were not done in duplicate, which every scientist does that. I mean, you don't do a single measurement, you do duplicate or triplicate. And no standard deviations were given, but yet they said that things got better without really doing any statistics. As I mentioned, not clear how dosing was done. And of course, we don't know of any adverse events. So I think that most of us, would agree that the methodology needed some improvement. And then, what were the results? Did it answer the question? Were the groups comparable? Did the treatment have an effect on the endpoint? Was their endpoint even relevant, and was the drug safe? How do you interpret the results, and what did this study add to our knowledge? And were there did the investigator talk about any caveats to the way they interpreted the results? Anything you should consider? Well, first of all, there was EMGs that were done before and after treatment. And the investigator claimed that there was improvement because some of the nerve damage went away. However, the EMGs were done by different clinicians at different institutions, and interpretation of EMG can be very subjective. And even on the first EMG, the nerve damage was not widespread. Again, sort of going towards that. Was this the right diagnosis? The variable, there was improvement in the ALSFRS score, but this could be within the realm of normal variability, and plateaus can occur. And the fact that this was not blinded, and some of the measures clearly are subjective and could be, this can cause bias. Okay. I know I'm getting a treatment. The investigator knows he's giving a treatment, and everybody wants to get better. That bias can introduce, especially in a subjective measure, improvement that really isn't there, and it really influences the objectivity. I try a little harder when you check my strength. I breathe a little bit deeper when you do my vital capacity, my breathing test. And I answer more optimistically when you ask me questions on the ALSFRS score. And as I mentioned before, as you walk, right, or you do an activity, the spasticity can get better. That's why we talk about stretching. Also, people could be on drugs, and the drug dosing could have changed over the course of the 6 months and influence your ability to walk. And so one of the things they showed was a gait exam that they claimed got better because after treatment, there weren't as many midline or crossing the midline hits. But that could have been stretching before the gait. It doesn't necessarily mean there was neuronal rescue, right? That doesn't necessarily mean the neurons were doing better. And, there's also training effects. So they talked about MoCA, and which is a cognitive exam. And you can, when you get the test enough, you kind of learn how to do it. And there's a training effect. You'll do better because you know what the questions are going to be. There was no adverse events. And I think one would say that this was exploratory at best and anecdotal. And the biomarkers are interesting, but it doesn't necessarily mean that ALS reversed. We don't know what the levels were before the person started the treatment or the trajectory or the natural variability in these measures, for instance, NFL. And there was no information in the methods on how they did the measurements, the number of replicates per time point. They had pre and post treatment 1 and treatment 2. There was no replication. It was single numbers. That wasn't done in duplicate or triplicate. So there's no standard deviations. And NFL is not disease specific. It can fluctuate. There's measurement variability. And a decrease does not necessarily mean that there's a reversal of ALS. Certainly that can be promising, but you can't claim that as showing reversal of ALS. HSP70 was the heat shock protein they measured, and it reflects heat stress response, but not ALS biology per se. And increases are normally expected when you heat somebody up. So it only shows that the treatment triggered that heat shock protein went up, but not necessarily that it helped the neurons. And then IL-10 is an inflammatory marker, and again, non-specific. And we don't know what it does normally in a person with ALS. We did have a longitudinal trajectory in an ALS patient that was untreated, or multiple people that weren't treated. And these also can be influenced by stress, temperature, infection, and again, don't necessarily correlate with disease progression. So to say that all of these changes mean that the neurons were better and treatment was working, it's a bit hard or tough to make that translation. And there was no direct ALS pathology markers. For instance, you could have done MUNI, motor unit number estimates, and shown preservation. But again, you need to know the natural history. And possibly one of the biggest things is that BTT Medical Institute, which is where this is being done, 3 out of the 4 authors have interest there. It's a big conflict. One was a full-time employee, and others, had monetary interest in this. There was only one person who had no competing interests. So, we can stop there for a minute. And Lyle, did you want to add anything?
No. I think one comment. So just so you guys know, Terry's going through some very specific science and some tests and some different chemicals, right? You don't have to know what this stuff is. I don't know what this stuff is. When we read papers, there's a lot of stuff we don't know. But what we do is we go through and we say, what was the comparison? What was the test, right? What was it that they're trying to answer, and what was their data? And if there's parts of it that we don't understand, we look them up. You can look them up easily nowadays in many different ways, and maybe we'll talk about that.
Yeah. And so for fun, I said, I'm going to go look at this BTT Medical Institute website, right? And see what's going on there. And, we all are familiar probably with ALS Untangled and the 10 red flags. So I decided to see how many red flags uh, were, you know, were on this website. And let me say that they pretty much hit everything. There may have been one I couldn't tell from the website. And when I tried to go in their chatbot to ask a question, the only way I could ask one was to get an appointment to see them for treatment. But the red flags, it was advertised as effective for multiple incurable conditions with different causes. And I have the list of all the things, the claims they made that they could treat. They had the videos of people who were cured, and they had so the only evidence of benefit was anecdotal in terms of their claims of reversal. There was no peer-reviewed evidence. What they had was articles, not that were published in peer-reviewed journals, but in newsletters and so forth by the lead investigator. There was no plan for presentation of outcomes for peer reviews, meetings that the team was going to be going to present their information. There was no randomized controlled trials that they had done with this. There was no blinding. It was all, N of ones. We don't know how they verified diagnoses. We certainly think that, I certainly think the ALS diagnosis may have been correct, but I have no way of knowing by what they put in the paper. And, the mechanism, this upregulation of heat shock protein, it may really be implausible because they have claims of cell regeneration. That's a nice big word, right? Getting better across multiple diseases with multiple pathologies. And of course, the conflict of interest is a pretty big one. And there's been no independent replication of these findings. So if we had a poll, I would ask this question, after hearing all this, would you A, get on the first flight to BTT Medical Institute and take out a bank loan to get the treatment? Or would you be awaiting a published double-blind placebo-controlled trial with validated endpoints and a documentation that the mechanism is relevant and had an effect? So with that, I'll simply say, and I hope you're all choosing B. But with that, I'll say, be informed and don't take everything at face value. And I actually get upset when I see this kind of thing because the real science, and there has been such real progress in treatment strategies and in assessments and remote assessments, as you know, from Everything ALS and all the clinical trials going on that really have valid reasons and a real background and a foundation to stand on. When I see this, it bothers me because there are going to be people who read this paper and who see their website and they're going to spend their life savings to go down there and get this stuff. Although I couldn't ask what it costs because they wouldn't let me do anything but schedule an appointment for treatment. With that, though, I will stop the slides and I guess we can start some discussion.
So Terry, I have two more quick slides before we do that.
Sure.
Can you see my screen again? Oh, wait
Yeah.
a minute. Wait a minute.
Not yet.
Not yet.
Yeah.
That was so great, Terry. Thank you for
Yes.
sharing.
I
Because-
hope so. I hope
Yeah.
everybody.
I
does
have
it.
to say, I was probably spending 10 hours a day when Peter was diagnosed with ALS. Maybe half an hour of that was useful because you can go through myriads of these traps and spend a lot of time on not relevant information. So thank you for that.
Okay, so to be very clear, we don't know. I mean, I want everything to work, right? I want that treatment that Terry just told us about to work. We all do. We're all on the same team. We want everything to work. And what we're saying is what's important to understand is how to be responsible when we read this information, right? Does a study or does some evidence that's presented actually prove what it's saying that it proves? And it's not that we're saying that the treatment doesn't work. We're saying that they need to do a trial. They need to prove it, right? And I wanted to sort of show, like, and I mentioned, that thing that Terry just presented had a lot of science in it. This is one, I don't know if you guys remember this one. This was, I guess, about a little over a year ago. A bunch of people emailed this to me. This was a press release, right, that came out. And we talk about press releases a lot too. Breakthrough that can halt the progression of ALS developed by Northeastern scientists. A major breakthrough in the treatment of ALS, known as, can potentially stop the disease in its tracks in as much as half of the cases in the US, a Northeastern scientist said. So somebody sent this to me. I was like, wow, that sounds cool. And I know some scientists at Northeastern University. And so what I did is what I hope you guys all do when, your relatives send you something on Facebook that says that Abraham Lincoln invented the microwave, right? You say, show me the source, right? Like what's the primary source? Where did this information come from? So when you read a press release, there's always a primary source. And I went and got it. And this is that article. Okay. And then I saw the article. I was like, oh my gosh, I know some of these scientists. These are like famous, smart people. Some of them are my heroes, like Bob Brown. Okay, Bob Brown discovered the SOD1 gene and really pioneered many of the ways that we, use like microRNAs and this sort of thing to silence gene mutations, discovered a lot of the mutations, figured out how to use the mice. And so I'm like, wow, so let's take a look at this article. And this article is got a lot of dense science in it, a lot of stuff I don't understand, a lot of complicated medicinal chemistry. That's okay. I can still read it and say, what's the comparison? What did they do? How did they prove it? And let me tell you what it doesn't say. It doesn't say they can halt ALS. In fact, this is a figure of the experiments that were done by Dr. Brown's lab in this paper. And the reason he was part of this study was because he's the expert in doing studies with the mice in the lab that have this mutation. This is a plot. It's the kind of thing that we're going to look at in future sessions, I think, a lot. Very common for the way that they do experiments in animals with a treatment. So you have, probability of survival. So at the beginning of the trial, 100% of the animals are alive. And then over time, they pass away. And this particular mouse tends to live, 130, 140 days. Okay. And so this kind of survival curve, like, in this particular animal, they're all alive and then they all sort of die around the same time. But here's the thing. This is the drug and this is the placebo. This is no drug. And there was no change. And it actually says, if you read it here, they did some stuff, and then based on these results to improve targeting, they increased the dose. No survival improvement was observed, right? So does that look like halt the progression of ALS? Of course not. Now, there was a second mouse model that's not a validated model, not one that anybody's ever used. It was very few mice, and the data was so questionable that they actually didn't include it in the main paper. It's in the supplementary data. But even that one just showed a trend. And so the point is and by the way, though, it's very fascinating science. It's science that I think is going to result in some real breakthroughs in the future and some real potential treatments. It's a really important paper, and it's a really good paper. This is irresponsible reporting on that paper. And so, you don't have to understand all the science in it to realize that. And that's kind of one of our points. And here's the thing, like the people who wrote that press release, or a lot of these press releases, like they're not bad people. They're not evil. They're not trying to, sell you snake oil, or I guess nowadays we call them essential oils because there is no snake oil. But if I'm a drug company and I spend hundreds of millions of dollars developing a drug, I want it to succeed. And I want to, if there's a sign there that it might succeed, I'm going to hype it because it's that's my perspective. It doesn't make me bad. But the way we talk about trials matters. And so this paper here, and you guys can look it up, these Dr. Finucane and Dr. Gilstrad, they collected a whole bunch of publications about this drug, donepezil, Aricept. And so this is a medicine that's used to treat Alzheimer's disease. And there were several late-phase trials, and most of them were industry-sponsored. A couple of them weren't. A couple of them were grassroots, not industry-sponsored trials. And when they looked at the data from all those trials was the same, literally the same. But the way they were talked about was very different. So the industry-sponsored trials said things like, oh, by the way, I should say the data was the same, and there was a statistically significant effect on this measure they used in the trial, in this scale to measure somebody's memory and cognition. The problem was the effect was so small that it was beyond the resolution that any person living with the disease or their family could even detect. And so the non-industry trial said this is a negative trial. And the industry trial said, oh, this is potentially interesting and maybe an early disease and as an adjunct. And so when people see this sort of thing, and when people see like the press release I just showed, we always say, oh, we need to punish the companies. At the end of the day, no. I mean, first of all, we'd like them to do a better job sometimes, some of them, of being more responsible, but that's like an infinite, never-ending mission. And so what we have to do instead is teach ourselves to read the stuff responsibly and to make our own judgments. And that's really the purpose of what we're doing. And the last thing before we get to questions, Terry just mentioned the 10 red flags. And I think most people on here know about this, but ALS Untangled, which we both participate in, which was started by Rick Bedlack at Duke to review off-label and alternative therapies. And you can go to this website, and there's a very standard way that we do this and produce this data, and they're very readable. And Rick's really done a tremendous job with this. But we came out with this thing. We went and looked through all of them, all the ones that had been done, and said, what do they have in common? And that's where the 10 red flags came from, right? And this is just sort of some things to have in your back pocket to be wary of. And Terry just went through them, but that a lot of these, essential oils, a lot of these treatments for which there may not be data is tended to share. So I invite you to go look at it. This is the paper for that. And now we can have some questions and talk about what you guys want to talk about.
I think Don has raised his hand.
It's Christian, are you going to moderate? I think you were making a Google Doc, right? Or Don, what did you want to?
Yes, we go ahead, Don.
Yeah, I really appreciate this. I want to make a couple of comments. You probably know this, that among the population of people with ALS, half of this is going over a lot of heads. I think I'm probably above average because I've had some background in statistics and things. By the way, my favorite statement about statistics is, if sufficiently tortured, statistics will confess to almost anything.
Yeah.
But my comment is, I know you're trying to say we are a team, and we are, but I think it's going to be a challenge to figure out how we can, especially those of us who have the disease but don't have a good medical background or statistical background or whatever.
Yeah.
Again, I think the other thing to keep in mind is that the risk toleration of us is much higher than most. So, we can dismiss an article like the one we just saw, which I'm wondering where in the world that was published. But anyway, there may be, most of us that participate in trials, we know they're very much long shots, and we wouldn't participate in them if we were saying, I'm sorry, we don't think. I think this is a 50-50 shot. We know it's not. So
thank you so much. And first of all, thank you from the bottom of my heart for participating in trials, right? Because that's the most important thing, and we really need it. And when you're in a trial, one of three things can happen. You can get better, you can stay the same, you can get worse, right? And honestly, if Vegas let us bet on clinical trials in ALS over the past 30 years, bet against them, because most of them haven't worked. Or at least it's not necessarily that they haven't worked. They may have worked for some people, but we don't have a good way to figure out who those people are because they haven't had rigorous biomarkers and good design, right? And we know that ALS is different for different people. But Don, one of the great things about this interconnected world we live in is those questions you're referring to that you're worried about having trouble with. Just ask, man. Like, we're always reachable and we're always happy to talk. And those are the kinds of things we can help with. And we definitely can talk to whether something is well designed and responsible, and what are the endpoints and why have they chosen them. And that's why we're a community, and that's why this is citizen-driven research.
Yeah.
And I'll also comment on the research, the PCORI type of research, where that's where we actually it's patient engaged comparative research, where we need people to be engaged as part of the stakeholders and part of the design of experiments. That's the whole point of PCORI, because some of the outcome measures, we were talking about how minimal an outcome measure, the Aricept, the donepezil, where there was literally an imperceptible difference to a person living with the disease, but yet it was statistically significant, so you could get registered as an approved drug. Well, in patient-engaged research, you tell us what's important as an outcome, what's important as quality of life, what you're willing to do. And so you're intimately engaged during the design of studies. So it's even not only participating in research as a quote subject, but also participating in the design. And everyone can participate in that way. And we'd love to have your input about what you think is important. Both Lyle and I are working on a project like that. I wanted to comment, how they got that published? Somebody was asking, how'd they get that published? Well, I think that the publication, if I'm not mistaken, is one of the predatory journals where you can pay to have your stuff published. And so, with a little bit of money, you can have it's sort of semi-peer reviewed, but if you pay, you get it published.
I think,
I get that email all the time, and I sent it to Lyle. I think, hey, all these people want us to publish. He said, do not respond,
Yeah. Well, I don't say, well, so let's be very clear. So first of all, I think we're going to do in one of these later sessions a little bit on the different kinds of publications, and what is a preprint, what is a peer-reviewed paper versus a non-peer-reviewed paper, what is a predatory journal. Don, one other thing you said, which is totally, really, really, we take it to heart. So this idea about, you have a fatal disease, right? And we get that. And we're right there with you. Like, I don't even know what it means to be an ALS expert if I can't, cure my patients, right? So I get it. Our job is not to tell you what to do, and I will never tell a patient, take this, don't take this. Our job is to educate you so that you can make an informed decision, right? Our job is to talk about the evidence for a therapy, the design of a trial, the risks, the benefits, the costs. If you hold me to the fire and say, if you were me, what do you take this? I'll probably answer you, but that's not my job. My job is to help you make an informed decision that aligns with your value system and what you want to do. And so, that's really the part of this that's important.
And we have a lot of questions that in the chat that Christian
do.
and Anusha are going to moderate. So
Yeah, we've had several, and I think that some of them are just as much topics unto themselves as just individual one-line questions. So I'm going to start with one of them, which is, when talking about the use of AI to summarize research, how do you have any tips? What have you found when you use it? Concerns or actually good experiences you've had, or even if there's if you've seen differences between how the different chat services use summarize research.
Yeah, so Terry knows this. I'm a nut for this stuff, and I like my Sunday night insomnia when I can't sleep. I'm like yelling at ChatGPT and having fun conversations. You have to be careful, right? So it's a really good tool. It really wants the large language models, which is what most of these are, have this tendency to want to make you happy and agree with you. And a lot of researchers are starting to use it. It will invent references that don't exist. It will invent people that don't exist. These are called hallucinations, right? It will basically infer what it thinks is likely to exist or to be there based on the information it can find. And you can ask it about that. And so what I would say is, just like we were talking about when somebody posts something on Facebook or a press release, you can ask any of these chat models, can you give me the reference for what you just said? Can you point me to the link to the information that you just gave me? And sometimes it will say, oops, sorry, there is no link. I made it up. I'm not kidding. It will say that.
Yeah.
And then it will say to you, would you like me going forward to not do that and to only provide information to you for which I can verify a reference? And you will say, yes, please do that. And 5 minutes later, it will do the same thing again, because it's not its fault. It's the way that these things are designed to infer information. Now, I'm biased, and so we just talked about not, don't understand when somebody has biases when they provide an answer to you. But I really like the EverythingALS AI chatbot, right? And I really like that we're making it better and trying to figure out how to do it better, because we're not training that chatbot on everything that's out there. We're training it on curated information, right? On stuff that we know is valid. So it's a good starting place. But the fact is, asking questions of AI, you can ask it for the references. Now, people are using AI more and more to write grants, to write papers, and this is a huge problem. And the industry is dealing with it in different ways and finding ways to detect this and stop this. But that's a whole nother topic.
Yeah.
Well, that's interesting you said that, because that's the next question, which is, is there one, maybe you can kind of talk to the problem of, AI in papers and, in references, and then maybe there are some reliable indicators or best practices that people can use to kind of look for that stuff.
Yeah. I mean, I think this would be an interesting thing for us to do as a whole session, but it's really the stuff I just said. It's, this stuff is evolving. If AI tells you something, make it give you the reference. You can have a conversation with it. You can ask it questions. I don't have any personal opinions about one being better than the other, except that I'm a cheap bastard, so I use the free ones more than the others. But I think it would be interesting to what I think we should probably do at a later session is, there is a growing literature on this stuff. And so we can find some recent papers and we can look at them together and see what we think.
It helps as a moderator to keep myself off mute. So there are some other good questions here. I'm going to switch topics a little bit. There was a comment that I think you could explore or just expound upon, which is somebody said, you can prove anything in a mouse model. And I know, Lyle, we've had that conversation before about the difference in results between mouse models and what goes to trial from there. So by all means, if you could explain more about that for folks.
Terry, you want to?
Yeah, I mean, this is so the first problem is misuse of the way the mouse model is used. The mouse models are, for instance, the SOD1 mouse model is a model of SOD1 disease, not sporadic ALS. So expecting changes in that model or treatments in that model to translate to people may be an overreach for the model. The second problem is that many people don't use the model properly because there's a lot of impact based on the background of the mouse model. The mouse model is based on putting abnormal SOD1 human genes into the mouse. You have 24 copies of it. And you put it on a genetic background, and there are different mouse strains, Black 6, SJL. And so if you're not careful about your mouse strain and the genetic background, and literally litter matching and gender matching, because gender makes a difference too in the mouse model, you can skew your results. So using the model properly and not and having the right expectations of what the model can tell you. The model has really certainly given us insight into mechanisms, and certainly the treatment of SOD was first, with tofersen, the antisense oligo that blocks the mutated SOD1 that we now know works in people, first worked in the animal model. And that's an expectation you can have, that you can take a treatment specifically for SOD1, works in the mouse, and then works in people with that mutation. So, and Lyle may want to add to those comments.
Yeah. I mean, so, okay. A tool, a model, right? It's only as good as how you use it, right? So, my kid when he was growing up loved snow, and like we'd measure how much snowfall. And he figured out that like if he held the ruler at an angle, it would measure deeper into the snow and it would be more snow. And so, that was not a good way to use his ruler to measure how much snow was there. And for most of the models in ALS, we have a whole lot of data and in fact guidelines on how to use them. One of the things you can use AI for is that if you're reading a paper and it's a paper and they've got let's say the SOD1 mouse, right? And they had 6 mice in one group and 6 mice in another. You can ask that AI model, can you provide me with the guidelines on how you're supposed to use this mice? Is 6 mice enough to tell if something works? Because, for example, in that model, we know you need to match sexes and you need to match litters, meaning the comparisons have to be between babies that are from the same mom and the same litter. And you have to have a certain number of mice to account for the statistics. And I don't remember all this stuff, like none of us do. But part of being a peer-reviewed publication is that when you submit an article to a journal, the journal is supposed to get experts in that stuff to review it. And they're the ones who are supposed to be checking, like, did they use this stain correctly in the microscope, or did they have enough mice in their mice experiments? And that really is the difference between a lot of quote unquote, high quality journals or low quality journals a lot of times, is how rigorous are they about finding qualified reviewers? But that's the point of peer review, is to figure that stuff out. Now, when you read an article and it's using a particular, let's say it's some kind of science, I don't know, atomic force microscopy that you never heard of, you don't know what it is. That is a thing where you can actually ask some of these conversational AIs. You can say, can you tell me about atomic force microscopy? How does it work? And I do that when I'm reading papers because it's like the world's greatest Encyclopedia Britannica. But
Right. Yeah, actually, kind of connected to that is, how do you identify or look up, maybe the AI is good for this, look up how reputable that journal is, or is there a telltale indicator can you pick one up? Let's say you're in the doctor's office in the lounge and there's publications out there. How do you know exactly what you're looking at right away? Are there telltale signs?
I mean, there's something called Beale's Index there's basically a couple flavors. And by the way, realize like it's not as much, it's who is the peer reviewer, right? And in this weird world we live in, we're all slave labor to do this for the journals. So anyway, that's a whole other topic. What you want to avoid are the so-called predatory journals. And if you guys look up predatory journals, give me a list of predatory journals. There are some services out there that log these and catalog them. And what it means to be a predatory journal is a pay for play, right? So if you're an academic, whether you get promoted, whether you get a raise, whether you keep your job depends on publishing. And some of these less savory journals, you can pay them a lot of money and you can, write in them that if you eat a raw potato in your shower standing on one foot, it'll make you, an opera singer. If you pay them enough money. So it's really avoiding the predatory journals. Beyond that, you got to think critically for yourself. You got to look, what was the comparison? Terry mentioned like a given model might be for one kind of ALS. We see this a lot, right? Somebody says, oh, I did an experiment and maybe it was in cells in a culture, but the cells they used were from somebody's liver. And I'm not kidding, by the way. And so is that really relevant to cells in your brain? And these are the kind of things that we're going to talk about in future sessions. How do you know? You don't have to memorize and learn all this stuff. You just have to know your marching orders and how to look at the comparisons.
Just to quickly follow up on that, because someone specifically asked about the journal Disease. Do you want to, if you could maybe make it here, do you know, like, is it a reputable journal? Should people?
Never heard of it.
Okay.
Is that the journal there was the article in the Terry that you just published?
No.
Disease sounds interesting, right? I'll bet I know what it's about.
Yeah.
There are these weird, some of the predatory journals, like they make plays on the famous journal, like, there's a journal called Neuron. And so there's a predatory journal called Neurons, or Cell and Cells. But again, like, you can ask also the AIs, is X a good journal? That's the kind of thing that AIs are good for. Is it a reliable?
What I just did.
Journal? Yeah.
I just did. It says it's peer reviewed and open access. So
yeah. So peer reviewed that's not enough information. You can say, tell me more. I want to know how, anyway. That's the kind of thing that LLMs are really good for. And you can ask it to go deeper and you can ask it, all those sorts of things. You can ask it what makes a good journal.
Yeah. Next question. We're switching gears a little bit. You mentioned, Terry, about disease mimics. as part of your explanation of that journal. Could you tell more what are disease mimics, and is it a set list? Is it something that is exactly? So if you could just describe more what mimics are when people hear that mentioned.
The first thing you want to do is rule anything else out that could cause motor neuron damage, right? And we all have our list of tests that we get, of things that can cause damage to motor neurons or look like they're damaging motor neurons. Hyperparathyroidism for some of us, hyperthyroidism, multifocal motor neuropathy, an autoimmune disease in which the myelin of the nerve, the insulation of the wire, if you will, can get attacked. It can look like ALS. So, heavy metals. Well, I'm not going to mention Lyme, but we all have our list of things and tests that we do to just make sure. Because personally, we all do things differently. Personally, I take making the diagnosis very seriously, and that I need to make sure that there's nothing else that could be mimicking. For instance, I get what's called an immune protein electrophoresis to make sure people don't have a monoclonal gammopathy that can lead to amyloid. That can, there's fancy words, but basically lead to a disorder that can damage the motor nerves and mimic a motor neuron disease. So, there's no set list. Everybody has their own kind of list, but it's really important to make sure this is a diagnosis of exclusion, and all of you who are living with ALS on this call have probably heard that and heard that from your clinician, that this is a diagnosis of exclusion. And, I personally will tell people I can never tell you I'm 100% certain until I have a biomarker of disease diagnosis. I can be 99% certain, but I can't be 100% certain. And, I'll let Lyle, because we don't necessarily match up, but
yeah, I mean, I don't think we don't match up. So, ALS is a clinical diagnosis, right? And so what does that mean? It means that you come see a neurologist like one of us and we examine you, and that's how we diagnose it. There is no, if you have diabetes, somebody can diagnose you by measuring your blood glucose and your hemoglobin A1C. If you have high blood pressure, somebody can measure your blood pressure, right? If you have a tumor, someone can see it. ALS depends on our clinical exam. And so when we talk about mimics, it's other things that clinically look like it. Things that cause painless progressive weakness that might spread around your body. And there's different diseases that do that, okay? Now, textbook ALS, upper and lower motor neuron signs, above and below the neck, certain things we see on exam. Textbook ALS is actually pretty easy to diagnose if you're an ALS doctor. And there's lots of studies that say we do a really good job at it. When somebody has ALS that didn't quite read the textbook, that's a little bit weird, that's when we think about what else could this be? And there's no one answer. It depends on what's unique about them. When a neurologist meets a patient, what we're trying to do is figure out where in the nervous system first is the problem. Is it nerve roots? Is it muscle? Is it brain? Is it spinal cord? Is it different places? How is it spreading? And then we think about the kinds of diseases that can affect those places and spread that way. And then we think about what are the ancillary tests we can order to confirm that, right? An imaging study, an EMG, a blood test. And there's a method that we go through to do this. Now, I always say to people living with this disease when I meet them, after the diagnosis is made, I'm not really that important. What's important is the multidisciplinary team that's going to take care of you. But I still follow along. And why am I, and one of the main reasons I'm following along is we never stop thinking about how could we be wrong? What else could be going on, right? But from a pragmatic standpoint, we want to check the boxes of certain other things that might be causing symptoms in you, and that depends on your presentation. And then we want to make sure we're doing everything we can to treat you if it's ALS, and certainly if we think it's ALS. And so, sometimes we'll see a patient who let's say we're 92% sure they have ALS, but we're not 100% sure, right? And we will still say to them, I really want you to take this medicine, Riazole. And ALS is a progressive disease, and it may not be obvious at the beginning that something is ALS, but over time it may become more obvious. And so while we're looking at these other things and doing these other tests, take this medicine, because if it's ALS, being on it early slows it down. And I don't have a time machine. So if I see you in 3 months and I say, oh shit, this is ALS. I wish I started you on this medicine 3 months ago. There's nothing I can do about it. But if I see you in 3 months and it turns out you have inclusion body myositis and not ALS, well, I can stop this medicine, right? I can stop Rysol and I can dance a jig with you and be so happy that I was wrong. And so from a pragmatic standpoint, what matters is checking the boxes, for each person of what might else be going on. But then at the same time, making sure that we're doing everything we can to treat you and help you slow down the disease.
Thanks, Lyle.
Thank you. So there's a lot of discussion about payments for therapies, for trials. I think it'd be helpful if maybe you could walk through, is that an immediate red flag? Are there situations in which that's actually a valid request? And kind of like how should people think about that?
Terry, you want to take it or me? Whatever you want.
ahead.
So, okay. I personally have an ethical issue with EAPs that charge my patients money. I think that a patient and their family paying for an unproven therapy is not the way we should do this. I don't have an answer for that right now, and there's a lot of people working on it. It goes way beyond just therapies, right? So I'm at Temple because of Terry, and she founded the ALS Hope Foundation, right? And the ALS Hope Foundation and the MD ALS Center of Hope was the first multidisciplinary clinic for ALS in this country. But as you guys know, they're all over the place now. And a lot of the big academic centers that have ALS clinics have a foundation attached to them. And it's like alphabet soup, right? Because there's so many different foundations. And why do they exist? They exist because somehow we need to subsidize the care of people living with this disease because insurance doesn't pay for it. Even though the standard of care in ALS is this multidisciplinary care, that's not paid for by insurance. And so that's why all these different foundations exist. And now the part of that I don't like is that a lot of these foundations, including ours, a lot of our fundraising is, event-based fundraising that is through the year, different kinds of fundraisers. And personally, and again, I don't have a solution, but the idea that it's my patient and their families and their friends that are subsidizing their own treatment isn't the way this should be. Now, of all the people I know, the person who's working to get this changed the most is Terry, because she's pioneered this, a lot of the groundwork behind this bill that called the ALS Better Cares Act that's been introduced in Congress right now, which for the first time is hopefully going to get a reimbursement code and payment for ALS multidisciplinary care. And she did that by a lot of diligent work looking at disparities in cost, disparities in care. Do you live near one of these centers? Do you not live near one of these centers? Looking at how much money is being lost everywhere. One of the reasons we don't have enough ALS doctors is that most hospitals won't let you start an ALS clinic because they're like, no way, man. You're going to take up a room for, 3 hours with a patient and bill for 20 minutes. And that's not a business model. So we have to solve a lot of this. How are we going to pay for stuff in ALS? When it comes to medicines, if I was living with ALS and somebody said this medicine will work for you, but you have to pay X amount of money, it would be hard for me not to pay that money. And so I feel like my role in this is to explain to somebody what's the data behind a given treatment, how much might it help, right? What are the risks? And different people have different value systems about that. I will say, though, I personally I wring my hands a lot. I don't think it's ethical to be having patients pay for EAP medicines.
Terry, anything to add on that one?
No, well, I totally agree with what Lyle said. I mean, and the same thing goes for these products like we talked about today in the paper. Show me the trial, show me the data, then insurance will cover it and people won't have to pay.
Yeah. Well, I'll stay with you on this because on the next question, because it was part of your presentation as well. You're mentioning double blind and placebo. Is it possible to double blind and placebo for treatments like the kind that are in that paper for let's say something there really is hope and there's something that's really kind of coming in from off of the typical radar, but may have been structured a little differently. Is it possible to double blind and placebo for those kinds of treatments?
Yeah, of course it is. I mean, first thing is the doctor can't know what the settings are on that fancy machine, and the settings can be set by somebody else, and neither the participant or the clinician who's administering it can, be blinded to what the settings are. They could be behind a screen that they can't see. And so they can be randomized to a group that gets the treatment, whatever the dosage was, and or a lesser treatment, or no dosing.
Mhm.
Yeah, Christian, it's actually much easier than you might think. I'll tell you what, the simple treatments are the ones that are actually maybe harder sometimes, and but all we can do is the best we can do. Like when I was a resident, we had a clinical trial that our institution was participating in of a stroke medicine that was given intravenously, right? And so somebody, when they came in for a stroke, they either got TPA, which is the clot buster drug that is the standard of care, or they got this other thing. They were randomized to this if they consented to it. And so one of these drugs was given as a one-time bolus. One of them was given as an infusion over a couple hours. And so the way that they tricked this was that they gave everybody a one-time bolus. One person got the drug, one person got a placebo, and then everybody got a bag over a couple hours, but one of the bags said drug, one didn't. And us, the residents who were doing this trial, we quickly realized we could tell who got drug and who got the TPA, because the tubing, it had a little color to it. It had a little shiny yellow color. And so we could see that. And so, these sorts of little tiny things make it hard, but it's really important to have blinded data. That the patient has to be blind and the doctor has to be blind.
Yeah, I mean, I've seen clinical trials where the adverse events with the drug were so great,
Mhm. Well, hopefully we're getting it to the point where, with the work we're doing with digital health technologies, with AI helping with maybe cloning and kind of digital twinning people, we might be able to have really good trials without placebo. I mean, that's the goal, certainly when engaging with the FDA, but it seems like just ignoring good standard operating procedure is not the way to go for these studies. So thank you for that. We've got a couple of additional questions that are a little bit more specific, but I think I'm going to kind of bleed it into a larger question. One is, the first part of it is, has there been any updated information regarding ALS4 that's been put out recently? And I would add, as just kind of to guide it to a larger topic as well, as how do guidances come out? Like, how do people, when it comes to ALS, keep up with new news, what's new guidance on a specific subtype or anything like that? Because it doesn't seem like there's as much of a governing body like there is, say, in cancer that kind of organizes it, or maybe there is and people just don't know where to look.
Well, so did you say ALS4? So you mean the type
ALS4.
of ALS, so the syntax mutation, right?
Yeah.
I mean, wow. So that's actually an example where a lot of the really exciting stuff is being led by grassroots patient organizations. There's a Facebook group. There's some other things. There's, again, in this data-heavy world we live in, there's never going to be some perfect source for this kind of thing, and it's always going to be changing and evolving. There certainly are some new therapies and some new recent publications related to syntax and that type of ALS. Right now there is no therapy that works for it as for others, but there is some exciting work specifically looking at the immune system and a particular kind of immune cells called T cells. There's a researcher at Mount Sinai. And if whoever asked that question wants to just send me an email, I can send you some papers about it. But again, we're all going to know when there's treatments that make a difference. I will say that is one example where there's been some recent advances in the science that look like they're going to bear fruit and maybe be able to repurpose some existing drugs.
Thank you.
We had another question about if there's any new research looking to the causation between 9/11 exposure and ALS. Because it's currently not considered a 9/11 associated disease, but has anyone done any sort of investigation into first responders, workers in that area?
So Terry, whatever you want, I can.
Go
So I
ahead.
mean, and in this world, in our world, the people who do most of the exposure and that kind of work is the CDC National ALS Registry, right? And they do a really amazing job with it. Now, there's this interesting phenomenon. with ALS and different sort of life events and exposures and illnesses. And this is the way I think about it. So you have to lose a certain number of your motor neurons before you actually have clinical weakness, right? And so by the time you become weak, a lot of the motor neurons have died. And so there's a period of sort of preclinical weakness before of disease before somebody has manifest actual weakness. And during that time, if something else happens to you, maybe you don't have the functional reserves to deal with it. And what I mean by that is, let's say somebody without ALS gets sick and lies in bed for 3 days because they're sick. And then after those 3 days, they get up and they want to go, go about their business, and they're weak, right? Because they've been lying in bed for 3 days. Well, if you're somebody living with ALS and you get sick for 3 days, or maybe you have a minor surgical procedure, or fall and injure yourself so you're walking a little bit differently, like you don't necessarily have the functional reserves to deal with that the way a normal person would. And so we think about that a lot when we hear, oh, my symptoms started after I had my knee surgery, or after I had COVID, or these kinds of things. So was it that it was actually triggered by those things, or was it that they had subclinical disease that was made manifest by some other insult to their body? And so that's always a challenge. Now, I don't know of any specific data about 9/11. It doesn't mean it's not out there. There certainly is a lot of data and research about different kinds of, job exposures. One of the things I do is I run a postmortem tissue core where people who've donated their brains and their spinal cords, we make sure that they get into the hands of researchers. And just in the past few weeks, we've gotten a researcher who is reaching out to us for specimens that are linked to some occupational exposures, military history, this kind of thing, for a hypothesis they have. So it's an active area of research. There's going to be a upcoming call for research proposals from the CDC National ALS Registry. They do this every year, specifically about evaluating risk factors for ALS, exposures, environment, life events, that kind of thing. And so there's a mechanism that researchers who are interested in this can get funded to do that research.
And it may be that it's really an interaction between your genetic background and your environmental exposures. And that's what makes it so complicated to study these things, and why we have so much conflicting evidence.
And actually, Terry, you make a great point. So, and on that, so, you might have seen studies that talk about, like, for example, that professional soccer players might have a higher rate of ALS, right? And so that in Europe, that there was this suggestion, I don't remember, it might have been in Italy, I think, that there were soccer players that had a higher rate of ALS.
Mhm.
And they were trying to figure out why. And they said, oh, maybe it's something in the grass, in the fields that that we treating the grass with. I said, well, hang on, because, they play other sports on those fields and they don't have a higher rate. And they said, well, maybe it's heading the ball and they're getting hit in the head a lot. And then, rugby players don't have a higher rate. And then somebody had this great idea. They said, well, maybe it's an ascertainment bias because there's a whole lot of ALS doctors in Italy and not as much in some other countries. And anyway, and maybe if somebody retires from playing soccer and they go home to wherever they live, maybe they don't get diagnosed. Don't know what happened with that. But then somebody realized something interesting, which is that if you're a professional soccer player in Italy, you've been playing soccer since you could walk. And maybe when you were 4 years old, somebody did some test on you, some fit test, and they said, okay, that kid's going to be a winger, that kid's a defender, that kid's a center mid, that kid's a sweep, whatever, striker. And that's what you play. And so maybe it's actually that the genetic predisposition that makes you a good professional soccer player, or a good, center mid, because it turned out there was a particular position that had the higher rate, also makes you have a higher risk of ALS. Maybe people who enlist in the military, maybe part of that is because of their genetic predisposition that makes them be a good soldier, right? You can play some funny math with this. So, like, C9orf is a genetic cause of ALS, and it's present in a certain percentage of people in the general population, right? And in ALS. So people of ALS, a certain percentage have C9orf mutations. If you're in the military, you have like 2 to 3 times more likely to have ALS. The percentage of those cases that are C9orf positive is the same as in the general population. So I think that means if you have a C9 mutation, you're you're more likely to join the military. Anyway, you can figure that out on a napkin and a pencil later. But I don't think that's true, by the way. But we do think that there's something about an interaction of environment and your genes. The studies you read about sometimes about, they find toxins in people's teeth, right? Have you seen these ones? Like they look in the molars and they find levels of certain metals. Well, that's great, but those toxins, those metals got into those teeth when people were babies, when they were infants, and in some cases even before they were born. And so maybe what it is a genetic predisposition to not handling metals the right way, as opposed to something causing ALS. So these are the kind of things that there's a lot of research on, and the CDC funds a lot of that research and does a lot of that research, but there's others too.
Thank you. I've got a really important question because it's really for the whole community to know with clarity about. If I'm progressing very slowly and could participate in a trial, but let me put it a different way. If I'm progressing very slowly, could participating in a clinical trial increase my progression rate?
Absolutely, right? So, but not so it could slow it down, it could make it go faster. And in fact, one of the other things that I think Terry's really a world expert in is nutrition and ALS. And she's partnered with EverythingALS for the nutrition app to try to figure out more about this. Where did that come from? Do you guys know? It came from, we had some trials in ALS of drugs that worked in mice, and then when we took them to people and tried them in trials, they actually sped up the disease. And we went back and looked and we said, what happened? How did they speed up the disease? And then we said, ooh, you know what? Those medicines had a side effect that they caused weight loss. And then when we looked further, we realized that losing weight seemed to speed up ALS and maintaining your weight seemed to slow it down. And that's where that whole thing came from. And so there's always a risk with, and we didn't see that in the mice. Why? Because you're force feeding them the same amount of food every day. There's always a risk in any clinical trial that something could accelerate the disease. It's not a different risk for a different person. The problem with slow progressors, which I think was the question in trials, is that you screw up the trial data because ALS is so variable, and we don't yet have good, rigorous tools to track individual people's rates the way that we would like to. We're working on it actually with EverythingALS. And so if a bunch of slow progressors happen to be in the placebo group, it makes it look like the placebo worked. I saw a patient this week in my own clinic who is just remarkably slow progressing. And I said to him, when I met you 3 years ago, if I had given you some magic drug, I'd be sitting here saying, oh, I'm the best doctor ever. Look at how slow you're progressing. But I didn't give you a magic drug and you're still progressing slowly.
That's really important for people to understand, but it's also, I think as they research trials and as they start to understand what's the right trial and look at protocols and those might be questions to ask. We certainly want to make sure it's not a chilling effect on trial enrollment as a whole, but it's certainly important for people to understand. So any advice on when reading about a trial, what to look at?
Your consent form should outline, you want to know when you're thinking about entering a trial, you want to know what's the drug, what's the mechanism, obviously. Does it seem reasonable? But what is it going to involve? How many visits? What do I do with those visits? What are the adverse events? What's known about the drug? What's known about that drug in, if it's a phase 3 trial from phase 1 and 2? It's a little harder if you're going into phase 1 because the whole purpose of a phase 1 trial is to make sure, I always say, to make sure you don't kill anyone. But, that's sort of tongue in cheek, but it's really to make sure that you have some dosing and you can look for any serious adverse events in a small group. And then, of course, a phase 2 trial is really supposed to look at safety and maybe get a hint of efficacy depending on your outcome measure. But it's really designed to collect more data on pharmacokinetics, how the drug behaves in the body, how the dose creates a level in the body, and then what side effects it has. And so always read over the side effects and understand these are all, as Lyle likes to say, experiments. We don't know what's going to but there is one thing I can say, and Rick always talks about this, that people who are in a trial do better, whether or not the drug works or not. And it's possibly because of the additional, interaction with the team, um, or maybe just people who are more into doing everything they can for themselves. But whatever the reason, participating in research, people do better. But don't kick yourself if you don't get into a trial, or because keep in mind, we don't know if it's going to work even, and there are adverse events. I mean, upcoming is Prime C's, something that's going to be coming up as a phase 3 trial. But both Cipro and Celebrex, which are the combo drug, have black box warnings. They're not the safest drugs, but you have to risk benefit, right? And that's important for you to understand what you're willing to put up with and what your risk benefit ratio is, and what you think would be meaningful as an outcome, and how that's going to impact your life, your caregiver's life, and your family if you participate.
Thank you. We've got a couple of final questions here. So Anusha's going to help us bring it home.
Yeah, so this last question is, when a neurologist tests for genetic causes for ALS, is there a routine or standard test that they run, or are they picking and choosing? Because, this person said that their doctor tested them, but how can she be sure that he tested for all of the possible genes?
I can take that. And by the way, just to quickly comment on that last thing too. So Terry mentioned something called PCORI earlier. PCORI stands for Patient-Centered Outcomes Research Institute. And what she didn't tell you is that she's spearheading this huge effort with all these organizations. And the whole point of it is to figure out what are the outcomes that matter to you guys, and how do we design trials that matter to you and decrease burden, and what does burden even mean, and what should we be measuring, and what do you wish your doctors didn't do that they seem to be interested in and you don't know why, and what would you like them to do that they don't do, and all those kinds of things. And so I invite you to look it up. Maybe Terry even, interestingly enough, we had just talked about how do we reach out to people to get their opinions with those surveys. I think we just found one answer to that. So
Yeah, focus group.
Yeah,
have
there's going to be some focus groups for that. So stay tuned for that. Now, the genetics of ALS. Okay. ALS is a rare disease. Genetic ALS is a super rare disease, right? If I'm a drug company and I'm going to spend millions and millions of dollars developing a drug, it's not a great business model when I've got 8 patients. And so once upon a time, we didn't test anybody not that long ago, we didn't test anybody for genetics routinely because it didn't change the way we treated you. We didn't have any therapies, right? It was expensive and hard to get the testing done. Now that we have these couple of therapies that seem to work for certain kinds of genetics, the plain fact is the industry, the pharma companies, they don't want to miss a customer. They don't want to miss somebody who might benefit from their drug. And so the genetic testing that we do in most clinics is sponsored testing, and it's sponsored by industry because for very legitimate reasons, it's important that they want to find everybody who might benefit. And so do I. I want to find all my patients who might benefit from a drug, right, or a trial. And so the sponsored testing we have is panels of drugs, and it's free, and it tests the mutations, panels of mutations, sorry. It tests the mutations that we know about in certain genes. That doesn't mean you might have a mutation that we don't know about or that hasn't ever even been discovered, right? But it's testing for the ones that we know about. And the reason we're able to do it, the reason it's economical, is because it's sponsored. And the reason it's sponsored is to catch everybody who might benefit from these trials or these therapies. Did that answer the question? I'm not sure.
I think so. I mean, I think
Yeah.
it's a constantly evolving landscape, right? With so many genes that are known to have maybe some degree of propensity involved. And
yeah. And we keep finding more genes, right? And while we say, wow, look at this, we found another gene, it's actually kind of bizarre, and maybe this is a topic for another day, weirdly frustrating that there can be so many different genes that cause this very specific disease. And there's some thoughts about that that I'm not going to talk about right now, but it's kind of strange, right? That all these different genes can cause this one disease. there are some companies you guys probably know, for example, this company Enlorum, which is designed to make custom gene therapies for very, very ultra rare mutations, like mutations that there's only 5 or 6 people that have. And they have an ALS program. It's called Silence ALS. It's spearheaded by Neil Schneider at Columbia. We're involved with it. Several other centers are as well. To custom make gene therapies. It costs about $2 million to make each one. Some of them can only be used by one person, right? $2 million for one person. Some of them can be used by more than that. Maybe we should do a session sometime where we talk about the different kinds of gene therapies and how they're developed.
It might be worth
Yeah.
it might be worth doing that along with genetic testing as well, because so much of this lands
that's a
right
good
now
idea.
in that variance of unknown significance, and what does that mean, and do neurologists even know what they're reading sometimes? So
And Christian, one last comment on that. So Terry has started a monthly clinic specifically for at-risk gene carriers, right? Because what happened was, because we started testing all these people for genetic mutations and we found them, whenever we find one, all the family members get tested. And so we're finding more and more people who have these mutations who don't have ALS. And those people want to know, what do I eat? What kind of exercise do I do? Where do I live? Does it matter, what kind of sleep I get? And we don't know the answers to those things. And we don't know how to do trials in those people yet either. And so Terry has actually started a clinic that is specifically tracking people who have these mutations who don't yet have ALS to help figure out some of that stuff.
and it's worth keeping track of when these tests get updated too, because new understanding comes out, and suddenly tests can get more precise and test for more genes. So it's not a one and done thing, right?
Yeah, I would just add as we close the comment that the genes that have been identified, and there's now over 40, have identified pathways. And I always say we're learning motor neuron biology, how motor neurons work gene by gene. And these pathways become important targets for treatment, and they are shared across different, mutations and sporadic ALS or ALS without a family history. And the other point that we should make is that we can now identify the genetic underpinning in 10% of ALS is familial. It runs in a family. In that 10%, about 70% of people, we can identify what the gene is with these panels that Lyle described. But there's still 30% that we can't, which means we're missing some genes. And it's an exciting time as we identify them and they identify pathways.
I think we've hit on a real topic there. Yeah.
Hey, Christian, questions that we didn't get to, if you compile them, and I mean, we're happy to do that offline.
We had a flurry of questions here at the end that we're going to compile, and we'll get to them as well. We're going to add them so the audience knows this is a series. It's not a one and done. So what we didn't get to tonight, Lyle and Terry, we're going to discuss internally, and we'll prioritize and package them for updates. And if you want to reach out and contact us, not wait for the next program, and you've got something really specific and maybe pressing that you want to ask, by all means, please contact us. You can write to Indu or me, our first name, and then at EverythingALS.org, or just write to info@EverythingALS.org, and we'll route the question from there. So
and by the way, guys, this is we want this to be what you want it to be. We work for you, right? That's why we're here. I mean, both Terry and I have devoted our lives to this disease, and we don't want to fail. We want to fix this damn thing, but we work for you. So we are very eager to hear about ideas about how you guys would like this done differently, better, what kind of topics are interesting to you. And this is going to evolve as we go.
You're not alone.
That's why we're here.
We're here.
Absolutely.
And also, and just as another little thing, I really, like I said, I love the fireside chats. They're just a much more open and just kind of talk about whatever you guys want to talk about. I join them. I encourage people, if you want to talk about these things more, join them. My only personal rule is on those, you have to call me Lyle. You can't call me Dr. Ostrow, because I'm not there as anybody's doctor.
Yeah, they are a tremendous resource for people. It's peer-to-peer. It's really the program for you, by you, as a community. So please join those whenever you have any kind of questions about living with ALS. So
thank you.
With that, thank you guys. Thank you, Terry and Lyle, as always. Wonderful topics, great explanations. We're really excited to continue the series. And as we mentioned, there will be, as part of this ongoing series, sometimes there will be a little bit of homework and pre-reads and things like that. So that'll be included in some of the communications that go out. We're going to put a little repository together too, if you ever want to go back and read some other materials. So with that, I'm going to give it back to Indu and McFinn.
Yeah, so I just wanted to announce that we are doing a new series, which starts on March 24th, and it'll be a monthly. It'll be focused on what we launched, EverythingAD.org, more focused on cognition. And so our speaker is going to be Sid O'Brien, very well-known Alzheimer researcher, and it'll be on March 24th, and you will get it on your email. And if you have anybody in your family, you know someone who've been diagnosed with Alzheimer's, please let them know, and you are welcome to come to this meeting as well. So it's about protect your brain is protecting your family. So it's going to be a lot of interesting cognition and Alzheimer treatments underway conversation. With that, thank you very much. And Lyle, Terry, so grateful. This is always, been my dream about getting people together, and this kind of made my day, made my year. So thank you, Terry and Lyle, for your time. We're really grateful for your friendship and this partnership.
Thank you.
And thank you, everyone, for joining us today.